Enzyme Substrate, a stop-motion studio film. Enzyme Substrate Complex. In an enzyme substrate complex, the substrate filled into the active site of the enzyme like a lock and key in a door. When the substrate, in this case the key, attaches to the enzyme, the door, a new product is created, the complex of the key and the door. The complex is held together by a hydrogen bond, electrical attraction, or a temporarily covalent bond. Competitive inhibition. In this case, the substrate is competing with an inhibitor of the same shape to form the enzyme substrate complex. When the inhibitor, the blue key, attaches with the door at the active site, it prevents the gold key from attaching. Therefore, it successfully acted as the inhibitor. Non-competitive inhibition. In non-competitive inhibition, rather than competing for the active site with the substrate, the inhibitor prevents the substrate from attaching by changing the shape of the active site. When the inhibitor attaches here, the keyhole is changed, which means the key can't fit and there will be no reaction. There is no way to attach because the active site has been changed. The key can only fit into a specific lock. Allosteric regulation. Allosteric regulation is very similar to non-competitive inhibition, but instead, it creates the opportunity for the substrate to attach rather than prevent it. When the allosteric modulator attaches, the active site is changed to fit the substrate. The black triangle here attaches back to the door, and the keyhole is fixed so the key can fit back in. Feedback inhibition. Feedback inhibition occurs when part of the substrate is left behind in the active site, which prevents anything else from attaching to the enzyme. In our case, part of the key broke off while the other part of the key is left in the door. This prevents the other key from attaching to our door. The part breaking off is feedback, which inhibits the new key from attaching. Denaturing of an enzyme. In the case of extreme temperature or an unusual pH, the enzyme will denature. This means that the enzyme is destroyed and obviously means there will be no more reactions that will be taking place. The fire here is very hot and it burns down the door.